Hello everyone, welcome you all in this video lecture. So in this video tutorial we will discuss about the ion exchange chromatography. So let's start with the introduction. So before moving to the ion exchange chromatography, let's understand that what is the chromatography. So basically the chromatography word is made up of two words which are chroma and graphene. Here the word chroma means color and the word graphene means to write. So basically the chromatography is considered for the separation of different types of molecules from a sample mixture. At first, the Russian botanist named as Michael Sweat coined the term chromatography in 1906. So Sweat used this method to separate the plant pigments, and that's why he used its name like chromatography. After that, the first analytical use of the chromatography was discovered by James and Martin in 1952, and they used this method for the use of gas chromatography for the analysis of fatty acid mixtures. Now, the chromatography is important technique in the field of biophysics and biochemistry. It is used for the separation and identification of the mixture. And this method enables the separation, identification and purification of the components which are found in the sample mixture. So basically the chromatography is used for the separation of different types of molecules or pigments present in the mixture. So basically the chromatography can be of different types. There are several chromatography methods which vary in their procedure and in their principle of working, like partition chromatography, adsorption chromatography, gas chromatography, ion exchange chromatography, and thin layer chromatography, and many others. So in this video lecture we will discuss about the ion exchange chromatography, its principle, procedure and functions in detail. So the ion exchange chromatography is also a type of column chromatography. We know that column chromatography is a type of chromatography which used columns which are filled with the mobile phase, and it is also considered as a type of liquid chromatography. So the ion exchange chromatography is used to analyze different types of ionic substances, which means that in the method of ion exchange chromatography, different ions or ionic substances can be separated, and the process allows the separation of ions or several polar molecules which are considered as ions on the basis of their affinity to the ion exchangers. So in this method different ion exchangers are used. We know that there are two types of ions named as cation, which are having positive charge in their structure, and another one are anions which are having negative charge. So the ion exchange chromatography is mostly used in the form of column chromatography. So now let's discuss about the principle on which the ion exchange chromatography method works. So in the case of the ion exchange chromatography, the sample compounds which are present in the sample mixture are separated by the electrostatic forces of the attraction. So the electrostatic forces are a type of force which are used by molecules to attach themselves. Now the segregation or separation of these molecules is based on the ion displacement theory. So we know that according to the ion displacement theory, the weaker ion is displaced by the stronger ion. Hence, the separation of these molecules depend on the type of the ion exchange resin used in the method. Now the charge group of the matrix can be positively or negatively charged, which are respectively named as cations and anions. So here when there is a suspended or any aqueous solution, these charge groups of the matrix will be surrounded by ions of the positive opposite charge. We know that opposite charges are always attract each other. Now here this is called as ion cloud, and these ions can be reversibly exchanged without changing the nature and properties of the matrix, and that's why it is called as ion exchangers, and the method is named as ion exchange chromatography. Now let's discuss about the instruments which are used in the ion exchange chromatography. So basically the method includes the use of pump, an injector, columns, a suppressor and some detectors. So the first instrument or the first equipment used in the ion exchange chromatography is pump. So the pump is considered as the most important instrument used in this method, and these have the function to provide a continuous constant flow of the element through the IC injector or any column or any detector. Now the another component is injector. So as we have discussed that it is a type of column chromatography. So here an injector is used to introduce the sample in the columns. So the sample introduction can be done by various ways, and the simplest method is to use any injection valve. So this injector is used for the sample introduction. The third instrument is columns. So here the columns may be stainless steel or maybe titanium, can be any inert plastic or maybe a glass. So these columns are made up of different materials, and they have the diameter from 2 millimeter to the 5 centimeter. The another component is suppressor, which is used to reduce the background conductivity of the chemicals which are used to elute samples from the ion exchange column, and this improves the conductivity measurement of the ions which are being tested. So this is used for the background conductivity. Now the another instrument used are detectors. So there are different types of detecting techniques, and these detectors are used to detect or to identify the separated molecules from the sample mixture, and after then a data system is also used for the calculation or for the analysis of the sample molecules or the molecules or pigments separated by this process. Now as we have discussed that the ion exchange resins are used in the process of ion exchange chromatography. So basically there are two types of ion exchange resins which can be used in this process. So these ion exchange resins are cation ion exchange resins, and the another one is named as anion exchange resin. So here the cation exchangers or anion exchangers are used, and the cation exchangers are those exchangers which possesses negatively charged group, and they are also called as acidic ion exchange materials. The another type is anionic exchangers which have positively charged group, and they are also called as basic ion exchange materials. So similarly the different types of resins used in this method. Now let's understand about the procedure or the working method of the ion exchange chromatography. So as we have discussed in our earlier slide that there are different types of components or instruments used in this method. So basically the separation is carried out mainly in columns with the packing of any ion exchanger, and we have discussed in our earlier slides that there are two types of ion exchangers used in this method. So the first step of the ion exchange chromatography consists of the selection of a suitable ion exchange resin which can be catabolic or anabolic in nature, and after then the another step includes preparation of the mobile phase. So basically this mobile phase used in this method can be a solvent, a solution, or a mixture of different types of solvents. So here after that, with the help of injector, this resin is introduced into the column, and first the column is filled with the ion exchanger, then the sample is applied followed by the buffer. So the process includes also a buffer solution. And there are different types of buffer like pyridine buffer, acetate buffer, phosphate buffers which are used in this method. After then, in the another step, the particles which have the high affinity for exchangers will come down to the column along with these buffers. So basically the separation of different ions or ionic molecules is depend upon the affinity or also considered as RF value for the ion exchangers. So after the separation, using corresponding buffers, these separates the tightly bound particles, and then with the help of different types of detectors, these separated molecules are detected or identified. So this is the procedure or steps which are used in the ion exchange chromatography. Now let's discuss about the applications or use of this method in industrial as well as in laboratories. So the ion exchange chromatography is used in deionization and softening of water. So as we have discussed that this method is used in the ions or ionic molecules. So that's why it is widely used for the softening of water. And the another application includes to analyze the amino acid mixtures. So we know that there are 20 principal amino acids which are found in humans as well as in other organelles from the blood serum, and also which can be found by the hydrolysis of proteins. So they are separated and identified by the method of ion exchange chromatography. Now it is also used to collect trace elements from the seawater and earth. So this is the most effective method for water purification also, and it is also used for the collecting resins which are used to collect trace material from seawater, and also to analyze lunar rocks and other rare trace elements found in the earth. So these are the applications of ion exchange chromatography. So basically it is the most efficient method for the separation of charged particles or inorganic ions which can be separated by this method. Besides these advantages and applications, there are several limitations of this method. The most major limitation is that it can be only used for the charged molecules or only separated the ionic molecules or ions by this method. And the another limitation is that it requires a buffer. So as we have discussed that there are different types of buffers used for this chromatography method. So these are some limitations also for this ion exchange chromatography. So in this video we have discussed about the ion exchange chromatography, its principle, procedure and applications. Thank you.